Hello, hello.
Yes, I can hear you.
Okay, it's time. Thank you all for coming to the fifth scheduled talk in gene therapy about making genes fit and to honor Dr. Ken Burns. Today, our speaker is Dr. Barie Carter. But before I introduce him, I just want to make two announcements. Dr. Song, whose talk was canceled due to the hurricane, will give his talk on November 11th, which is a Monday, but it will be at 12 o'clock. That's the only time we could get the room. So please come for that, November 11th. And then next week's talk, Dr. Terry Flott, is at 1 o'clock, not at 1:30. Please come at 1 because he has to be at a meeting at 2 o'clock. So we had to accommodate the speaker. So I hope it'll be okay for you guys. Okay, now I would like to introduce the speaker for today. It's Dr. Barie Carter, and he is, his long-term research interests are in biology of viruses and the clinical development of gene therapy. At NIH, he studied the adeno-associated virus biology and genome structure and expression. And he developed the earliest AAV vectors, including the first filed and issued patent on an AAV vector. In the biotechnology industry, he led the clinical development of these vectors, and at Targeted Genetics, the company, he initiated in 1994 the first clinical trials of an AAV vector. At BioMarine, he established and led the gene therapy program, which resulted in the recent approval of an AAV gene therapy for hemophilia A. He's the past president of the American Society for Gene and Cell Therapy. He has served on the advisory committee to the director of the NIH and on many other advisory and review committees for NIH, for the FDA, the Cystic Fibrosis Foundation, the New Zealand Medical Research Council, and the School of Public Health, University of Washington. Today, he comes to us from California, so the speaker will be on Zoom. Please welcome Dr. Barie Carter.
Thank you, Push. But first of all, I'm not in California. I actually am in Seattle. I live in Seattle.
I'm sorry.
It's okay. And all the time I was working at BioMarine, I just commuted back and forward. So anyway, I'm in Seattle. Now, first of all, I want to thank you very much for the invitation to talk and to honor Ken, who was a very long-term friend, scientific colleague, and a great person. And so what I want to do today is talk about two aspects of development of gene therapy with AAV. And I understand you've had some talks on AAV already, so you all have some general background. And I want to talk, the title From ITRs to Hemophilia is meant to reflect, I want to talk about some very early work on ITRs, because that was work that Ken and I did in parallel and jointly, and that was a long time ago, and it was great fun, and it was sort of early stage work on AAV. And then I want to end up talking about the development of the hemophilia gene therapy. So now my slide doesn't want to advance. I don't know why. Why is that?
Again, click it again. Try now.
I should hit the return button, I thought would advance it, or is it the forward button?
The up and down button.
Oh, the up and down button. I can't see a button to press, boy.
Yeah, I click on the screen, and once I click, you should be able to adjust the slides. So you should be able to do it.
Oh, I see now. So what am I seeing on the screen? I see. I got it. The forward button here. Okay, got it. Thank you very much. So this, I think you're aware of, that AAV, adeno-associated virus, is a small protein capsid, and inside that is a single strand of DNA that's about 5 kilobases long. And the way we normally schematically draw the AAV DNA is shown on the left there. It's that white line, and as you can see, it's got these two sort of T-shaped folds at the end, and those actually represent what are called inverted terminal repeats. And it actually turns out that it's the same sequence at both ends. They are critical for amplifying AAV DNA, for replicating it, and they're also the critical signals for inserting that strand into the capsid. And so when we make vectors, and as I think you've probably heard already, using AAV as a gene delivery system for gene therapy, or as it's called, a vector, they're actually based on the wild type AAV. And the top schematic here with the orange colored boxes represents a schematic drawing of the normal wild type AAV. And in the left half of the genome, it has codes for a family of proteins called REP that are important for the replication and packaging. And on the right, it encodes for the capsid proteins that make that capsid. And there's another little protein called AAP that's important in the assembly. When we want to make a vector, we take out all of those gene sequences from AAV, so there's no viral genes left in the sequence, and we add in the gene of interest, which what I will eventually talk about will be for hemophilia. And so the only part of the AAV DNA that remains in the vector are these ITRs at the end. And they're only about 145 nucleotides long each. So there's only something like less than 400 nucleotides of AAV DNA that's present in a vector genome. And so let's talk about the origin of those, because I think large numbers of people using AAV vectors nowadays may not even know really how those were evolved. And it allows me to talk about my early interactions with Ken Burns. So I don't know if anybody's told you yet, but Ken, in 1969, was at NIH and working with Jim Rose. And they did a very critical experiment, which Ken actually devised and thought of, to show that AAV packages a single strand of DNA, but strands of complementary sense in different particles. And it was actually quite a classic experiment at the time. And it certainly got my attention. And in 1970, I arrived at NIH to work with Jim Rose in his lab and to start working on AAV. Ken had just left and gone up the road to Baltimore back to Johns Hopkins to a faculty position. We hadn't met before, but I knew who he was from the work. We'd both worked on molecular biology of bacteriophages in the 60s, and I had read his papers. But very soon after that, I met Ken, and that led to 50 years or more than 50 years, I think, of really terrific scientific and social interactions, a very valued friendship. And so that was something which he was largely responsible for getting me into AAV in the first place. And that continued over a long time. And in the 1970s, we both he was at Hopkins and then moved to Florida somewhere in there. And I was at NIH. We worked on the structure of the AAV DNA and particularly realized that it had a sort of unusual structure at the ends. And I think you've probably been told that AAV was only discovered in 1995 at the University of Pittsburgh and at NIH. So we knew very little about AAV. So this was very early days. And ultimately, the whole description of these ITRs, and by 1980 was Ken and his lab did a beautiful piece of work to actually describe the complete sequence of that DNA. And if I just go to the next slide, I don't want to go through the specifics of how this happened, but the whole story of the structure of AAV ITRs, the structure of them, not the function, evolved over 10 years, actually, from about 1970 to 1980. You might wonder why it took so long. Well, part of it was technology didn't exist. In the early 70s, we both were using very early types of technology, chromatography, enzyme digestions at the ends, and electron microscopy. And that clearly pointed out that there were repeated sequences at either end that could interact with each other. In the middle of the 1970s, we got restriction endonucleases, and these are enzymes that can cut DNA at very specific sites, and were really important to begin mapping DNA genomes, and so we could do that. And in fact, in the midst of that, we actually published a joint paper together in 1976, and that work led to showing that we could show that the ends had some structure that could fold up. And then around a little later in the 1970s, DNA sequencing techniques became available. Two sets of techniques from the US and from Europe. And Ken's lab did a nice job with, it was Ed Lusby, I think, at the time. And by this stage, he was in Florida, did the complete sequence of the ITR, which clearly showed that the sequence actually is a palindrome and it can fold on itself and form this structure. And then a couple later, his lab, along with Arun Srivastava, actually provided a complete sequence of AAV2 DNA. So now we had the whole full sequence, and that was critical because now we could start thinking about analyzing this in much more detail. And we actually could start thinking about, could we turn AAV into a gene delivery system, given that the parental virus is not pathogenic and doesn't cause disease? So it would seem like a good, safe place to start. However, there was a roadblock, and that was that there were guidelines. NIH had guidelines for working with recombinant DNA. And at the time, up until 1980, these guidelines required that if you wanted to do this with human viruses or DNA from any human virus, you had to do it in a containment system, which was called P4 then, or BL4. This basically means a completely isolated, everybody is working in space suits, and all the experimental work is done in big gloved hoods. These facilities existed essentially nowhere, except maybe at Fort Detrick, the Army had one, and maybe on Plum Island. But fortunately, in 1980, Ken became a member of the NIH Recombinant Advisory Committee that sort of oversaw these guidelines. He got together about a dozen of us who were working on these viruses, and we talked about this and came with a recommendation that we could reduce the containment. Really only needed to be the normal containment we used when working with the parental viruses, which is a standard lab and a biosafety hood. At the time was called P2. So the group recommended that, and almost immediately the NIH Recombinant Advisory Committee adopted that recommendation. And so once that was done, we could start making molecular clones of AAV, and was really off to the races. And the reason I go through this is I think, is a really critical role that Ken play in certainly in allowing us to get on with AAV as a gene therapy vector. And I don't think this is generally recognized anywhere, but it was a really crucial thing that he provided. So pretty soon after that, the Florida group, led by Nick Musichka, and along with Ken and Jude Samorski, produced molecular clones of AAV. These are where you put the entire genome into a plasmid, a bacterial plasmid, and you can amplify it up. They did that in one set of plasmids, and my lab did that at NIH. And so now we had plasmid clones of AAV, and it turned out remarkably that when we had these clones, what's shown on the top here is a schematic representation called wild type AAV. Is if you take one DNA with these molecular clones, put it in a cell, and you have to provide the helper virus, which I presume you've heard about is normally adenovirus or adenovirus genes. If you put that on the cell, what comes out is the AAV genome excises and replicates and makes infectious AAV. And that immediately told us how we could make vectors, because in the bottom part, what we do is in the molecular clone, we take out all the rep and cap genes of AAV, and we put in the gene of interest. We introduce that into the cell, and we also provide to the cell, you still have to provide those rep and cap genes. And there's a number of different ways to do that. But if you do all that, then what you get out is the particles showing with the green DNA in there. You get particles that have the vector genome in them. And so these are AAV gene therapy vectors. So that, interestingly, was exactly, we made vectors, and both Nick Musichka's group made vectors in Florida, and we made vectors at NIH and showed that we could express what were basically called reporter genes at the time. And it's interesting to note that both groups published our work exactly 40 years ago in October of 1984. Once we had that, we could think about clinical development. We began thinking about gene therapy vectors. And at NIH, because the Cystic Fibrosis Foundation was just down the road and my institute had an interest in that, we started thinking about cystic fibrosis. And one of the reasons was that the CF gene, as you probably know, was cloned in 1989. And the lung is a directly accessible organ via the airway. So it was thought at the time this was a great target for a gene therapy. And at the same time, Terry Flott, who you're going to hear from next week, had done his pediatric pulmonary residency at Johns Hopkins. He came down to my lab and wanted to work on viruses and gene therapy, and he was actually funded by the CF Foundation to do this. And we started working on developing delivery to the lung with the idea of going after cystic fibrosis. By 1992, I had moved to Targeted Genetics, which was a startup gene therapy company in Seattle, and Terry was back at Johns Hopkins. And we decided that we should move ahead into the clinic and see what we could achieve. There were all sorts of challenges because there had been no prior animal studies with AAV vectors. There was no, GMP manufacturing system. There was no actual regulatory pathway for AAV development. And we were the very first group who talked to the FDA about developing gene therapy and what would be the requirements and how would we go about it. Interestingly, things move really surprisingly quickly. And actually, I think it was 1995, at Targeted Genetics, we had produced the vectors and we began the first AAV, or first clinical trial of any AAV vector. That was at Thanksgiving in 95. And the first patient was actually treated at the University of Florida in Gainesville because Terry Flott by then had moved down there. And he was the principal investigator on the trial. Ultimately, CF didn't pan out. It turns out there are a bunch of other reasons why it's not a particularly good target for gene therapy, but it taught us an enormous amount. And so here we are now, about 30 years later. There are actually 7 AAV gene therapy products that have been approved, and I'm not going to talk about all of them. But I will talk about the one second from the bottom. It's called Roctavian, and it's the hemophilia A gene therapy produced from BioMarin. And that's actually the largest patient population of any of these, I think, for which gene therapy is being developed. So if we jump ahead, there began to be a lot of people working on AAV gene therapies but there was a really important paper published in 2011, at the end of 2011, in New England Journal. And this was describing actually a proof of concept of an AAV gene therapy for hemophilia B from the first author was Amit Nathwani, who's at the University College in London, or University of London, England, and from Andrew Davidoff and his group at St. Jude's in Memphis in Tennessee. And what they showed was with an AAV vector that contained a gene for hemophilia B that I'll mention in a moment, they could deliver this by peripheral vein infusion and show that they could stop the bleeding phenotype in hemophilia B patients, and they did not need to use a lot of their protein prophylaxis. The critical part here was to deliver it systemically by peripheral vein infusion, because then it goes mostly to the liver, and the liver is where the hemophilia factors are normally made. So that set us on, and around about this time, I had joined BioMarin because they wanted to get it, do gene therapy. They were enzyme replacement company, but nonetheless thought that gene therapy was important as well. They didn't have anybody who knew about gene therapy, so they convinced me to join the company. We looked around a lot of targets, and we actually ended up licensing some technology from Amit Nathwani's lab in London, which was the basis for starting off to look at a gene therapy for hemophilia A. So before we talk gene therapy, let me just summarize about some things about hemophilia. As you know, hemophilia is a bleeding disorder where your blood doesn't clot properly. And this slide up here, which is ironically described, labels the coagulation cascade simplified, is that the clotting cascade for blood is actually enormously complicated. There's a whole string of factors that interact successfully in successive steps before a clot forms. But the two things, I don't know if you can see my point here. There are two proteins that are quite critical in this. One is called factor 8. It's this one over here. And the other one's called factor 9, which is this one over here. And at the critical part of this pathway, these two proteins, factors, have to interact. And if they don't, if either one is missing, then you cannot clot blood. You have a bleeding disorder and you have hemophilia. So there are actually two types of hemophilia. There's hemophilia A, and that's a deficiency in the clotting factor, factor 8. And there's hemophilia B, which is in which you're missing the clotting factor, factor 9. Hemophilia A is about fourfold more prevalent. It's about 1 in 5,000 male births, and the other one about 1 in 25,000. But there's a pretty large number of patients worldwide, and as you know, I'm sure you all know that hemophilia is a difficult disease to deal with. Interestingly, both diseases are essentially the same because missing either factor causes the same set of symptoms and the same clinical problems. So at Barie, we decided to do hemophilia A much because there's a bigger need than for hemophilia B. And the other things that I need to point out is that hemophilia A is an X-linked disorder. So it's a disease that affects males because they only get one X chromosome. And if you have the mutation, then you'll end up with a hemophilia. A famous carrier of hemophilia was actually Queen Victoria in England in the late 1800s. And in fact, the royal families throughout Europe had hemophilia, including the Russian Tsars. One of the Tsar's sons had hemophilia. There are several sort of levels of severity of the disease. If patients who have less than 1% normal amount of the factor 8 or factor, in case of hemophilia A, have what's called severe disease, and they get really substantial spontaneous bleeds, and I'll talk about joint problems, etc. Patients with a bit over 1% have a moderate version of the disease. Spontaneous bleeds are less rare, but excessive bleeding can still occur from quite minor trauma. And then a mild version, those who have something somewhat over 5% or perhaps 10% of the normal level, still require treatment for dental procedures and surgery, but have somewhat less of an issue with spontaneous bleeds or increased bleeding time. So in thinking about, and this is even though patients are treated with protein prophylaxis for them, you can have really serious problems. And this is an example of what happens. The problem is, even though you have a low level of blood, you get spontaneous bleeds into joints, particularly just with a normal movement of joints, and little capillaries bleed off and on. And you can get these terrific hemathroses in joints. And in fact, you can get to the stage where joints have to be replaced. So there's a real need for over and above the current protein therapy for hemophilia to try and avoid this type of clinical problem. And over time, let me just click all of these on because it's so originally, up until about 1960, there was essentially no treatment for hemophilia other than if you got a bleed, you had to go get hospitalized and be transfused. Once factor 8 and factor 9 were identified in blood, then there began to be concentrates of these factors. They were simply plasma concentrates which included these factors, and those were used, in particular, they could be used at home to infuse into hemophilia patients. But one of I'm sure you remember the terrible things that happened to hemophilia patients who got these crude lyses were that they also suddenly got infected with what at the time were unknown viruses, including HIV and hepatitis. And so a large proportion of older hemophiliacs are still HIV positive. And this was a major problem. Fortunately, in the mid-90s, certainly for factor VIII, the gene was cloned, and now that allowed production of recombinant factor VIII, which was much higher purity and not contaminated with these viruses. And so that's being generally used for factor VIII, and the people get infusions of or self-injection about 2 or 3 times a week. And over time, some of the half-life of those proteins is not very long. They dissipate rather quickly in the blood, so you have to take them very frequently. So the idea was that perhaps with a gene therapy, we could do a one-time delivery of a vector, and we could make over a prolonged time a constant level of the blood. This is a note. So I said that factor VIII gene was identified and cloned, and the structure of the protein was determined. This also happened, interestingly enough, in 1994. There was a tour de force of 3 papers published in Nature from the group Genentech, in which they cloned the gene, they characterized it, they managed to express the protein and showed that the protein was active. And this last one, it's called Structure of Human Factor VIII. One of the main people in this group was a guy called Gordon Vehar, who was the first author on this paper. And when I got to Barie, Gordon Vehar, who was there then, he's still there now. So when we launched into doing gene therapy for hemophilia there, it was terrific to have a really superb biochemist who really knew the ins and outs of factor VIII protein. Interestingly enough, that was in November of 1994, and that's just 1 month after the first publications I described on AAV vectors. So it's an interesting set of coincidences, these things. Years later, we combined 2 of them at when I got to Barie. And so, as I've mentioned, the real goal of gene therapy is to try to avoid having to do frequent injections. And the other issue is these proteins, you inject them, you get a peak, but then it goes down to a trough, and it can go way below 1% before you give the next dose. And so the difficulty is that whenever it's down in those troughs, you can have minor bleeds, and you still have ongoing problems. And the concept, and this is true for a lot of gene therapies at enzyme deficiencies, where you can try and have a continuous level and avoid these low troughs. And for hemophilia, even with the protein prophylaxis, the risk of injury is always there, and it's a really difficult life burden to manage their protein prophylaxis. And if you could avoid much of that, it's a pretty big paradigm shift in their life. So, and this chart here shows the amount of bleeding per year versus the levels of factor VIII. And it's very clear, the lower the factor VIII levels in hemophilia, the much more frequent serious bleeds and things are. And so what it shows, leads to us show this, that if we can get to about 5, this says level of activity, but it's also the percent of normal level, around 5 or 10% level, you could do a very good job of really lowering much of that bleeding. And so the original goal of gene therapy was to try to get constant levels that would be above about at least 5% of normal. So the vector that we constructed at Barie is this one. It looks like all other AAV vectors. It's got those ITRs at either end, and inserted in that is a gene for factor VIII. Now, the issue is the full gene for factor VIII is too big to fit in an AAV vector, because the size limit is about 5 kilobases for these vectors. But as it turns out, when factor VIII is made in the course of being secreted out of the liver into blood, a middle section of it called the B domain is actually deleted. And so the protein that's out in blood and that functions for clotting is a smaller gene, and the gene for that it fits inside AAV quite nicely. And so we used the gene for that, or the cDNA for that in the AAV vector. And what this vector is designed to do is to make that what's called the B-deleted protein, and it's exactly the same amino acid sequence as is present in Refacto or Xintha. And these are the the recombinant proteins for factor VIII that are actually used for prophylaxis. And so regulatory agencies like the FDA were like that, because they said, right, so at least the protein you're going to express we know about, and we know exactly what it does as a recombinant, and it's been used in large numbers of patients. So we're making exactly the same thing as the protein prophylaxis, but by using hopefully a one-time delivery of the vector. In this case, the AAV capsid that we used here is one called serotype 5 that was originally discovered from humans in Germany a long time ago. It turns out that this is important for two reasons. It really has when delivered intravenously, goes largely to the liver, which is where we would like it to go. The other thing it turns out is that the big issue with AAV vectors is they have a protein capsid. And so once you've delivered them, you're going to have an antibody response against the capsid. You're essentially vaccinated. So you can't give these vectors repeatedly. So you have to get it right the first time. And more importantly, if you've been exposed to these vectors or one given serotype or another in somewhere in the past, then you may already have pre-existing antibodies which would block that vector from working. And the common ones in AAV are serotypes 2 and 3 and some others. As it turns out, AAV5 is about the least common pre-existing antibody present in human populations in most parts of the world. And so it is something like maybe 20% or so of subjects can't be treated for with an AAV5 vector. You have to use something different. Nevertheless, we went ahead with this, and I'm not going to go through all of the animal data and preclinical data which we did, but and also BioMarin had set up an enormous manufacturing system. It's a big making large doses of these things because the doses have to be high, and their experience in producing recombinant proteins was enormously useful in now producing large amounts of an AAV vector. But the first clinical trial which we did, and as you know, clinical trials in humans are normally done in phase 1, phase 2, and then phase 3, or sometimes phase 1 and 2 are combined as here. And that vector was tested in patients with hemophilia A in a phase 1/2 trial. There were about 15 or 16 subjects, and we actually used 2 different doses. This is a dose here. It's doesn't want to. The dose at the top shows levels of factor VIII activity over a period across there is weeks, and so it's up to 156 weeks. And you can see that the amount of factor VIII in the blood after a single dose of the vector rises for the first 3-4 months, and then is pretty steady, but trails off a bit over the course of 3 years. The bottom panel is using a little bit lower dose, and the levels are good, but they're not perhaps quite as good. The 2 different colored bars there are simply 2 different ways of assaying the level of factor VIII. They essentially show the same thing. And on this scale, 100 is around the normal level. And so you can see that most of these got up to around normal levels, certainly in the first year, and were maintained certainly at levels that would be mild disease. And importantly, if we look here, the top left panel where the red and the blue slides are, that's just as mean or average, mean or median rather. This shows in the top panel A at the higher dose, the number of annual bleeds, of serious bleeds, prior to them being treated. And then you can see afterwards at year 1, year 2, or year 3, that number is decreased to somewhere close to 0. The lower on the left, panel C, shows at the same dose the amount of factor VIII protein prophylaxis which these patients needed after infusion of the vector, and you can see that that's almost eliminated as well. It's gone very low, and similar results, almost as good at the lower dose. So that was in that trial. Those patients are now being followed out 5 years, and they've still maintained production of factor VIII, even though the levels have dropped somewhat, and still have substantial reduction in bleeding events, and mostly are still not using prophylactic factor VIII. The gene therapy is pretty safe. It was well tolerated. The one thing that does happen, and this turns out to be a phenomenon of most AAV vectors, particularly used at quite high doses and delivered systemically, is that there tend to be some elevations of liver enzymes, ALTs. Mostly, certainly the ones we did with AAV5, these were not, they were listed as grade 1. They're never more than 2% or 2 times the upper limit of normal, which is a relatively mild liver increase. Nevertheless, it's a liver enzyme increase. And this is treated and can be treated with, of course, corticosteroids, which are generally pretty well tolerated. And so typically now you can expect when delivering this vector to also be tracking them for several first few months. And if there are these moderate rises in liver enzymes, treat with corticosteroids. There have been some other trials where even higher doses of AAV have actually led to quite serious liver enzyme rises. So this is one particular limitation of AAV vectors you need to be careful about. So having done, on the basis of that, BioMarin launched phase 3 clinical trial for hemophilia. And this was done in the UK and several European countries, ultimately in the US as well. And this trial ultimately enrolled 134 patients, all of whom had severe hemophilia, so they're all below that 1% level, and with a couple of requirements, one being that they did not have any pre-existing antibody to AAV5 serotype, and that they did not have inhibitor formation against factor VIII. Some hemophiliacs have develop actually inhibitors against factor VIII. It's an antibody to factor VIII. All of these patients, as I'm going to show you, showed, well, and again, there were some elevations in liver enzymes, and those were well controlled with glucocorticoids. By this time, the vector is called Veloctagene roxaparvovec, which is a sort of official FDA-approved name of the vector, and parvovec means it's a parvo vector, and then the other parts are the specifics for this vector. And all of these subjects showed enormous, or virtually all of them showed very reduced bleeds and greatly reduced use of factor VIII concentrates. In fact, most of them didn't need factor VIII prophylaxis. And I think on the next slide, so here's again the levels of factor VIII activity in blood. The top panel shows all 132 patients, I think it was, over the first year, and you can see they've all got levels of somewhere around, averaging around 40 or 50, which is about 40 or 50% of the normal level. And the bottom panel at the time when this was published shows 17 of the subjects who'd gone out over 2 years. And as you can see, they still maintain factor VIII expression in the blood. It decreases a bit, but it's still up around at least 20%. And this certainly is in the mild range. Similarly, again, looking at amount of factor VIII prophylaxis, which was used after the treatment, and it's essentially zeroed out. The number of infusions of factor VIII required after the treatment, again, is very close to zero from what had been 128, 135 is the normal number of infusions a year. That's 2 or 3 times weekly. And similarly with transient bleeding episodes, dropped quite dramatically again, and very few of those. So this essentially showed the same results or amplified the results from the original phase 1 trial, now in a much larger patient trial. And again, this was a subsequent publication in 2023 just provided more data on this and much more safety data. And this was out over nearly full 2 years of all of the subjects. The reason for this was the FDA wanted to see absolutely full follow-up for at least 2 years on all of the subjects in the trial. And so that's the second publication. So on the basis of this, and the conclusions from this phase 3 trial is that there was clearly durability of factor VIII activity and bleeding reduction, and the safety profile was pretty good for at least 2 years. And the looking at the risk of joint bleeding shows that the relationship between the amount of factor VIII activity and the bleeding was similar to that with the epidemiologic data. And so essentially these serious patients had been converted to certainly mild and in some cases moderate hemophilia A. Consequently, the European Commission granted marketing authorization. It was actually conditional, but it's marketing authorization in August of 2022 for adults with severe hemophilia, and importantly for the adults without antibodies to AAV5. And actually also gave it orphan drug designation, which means that for an AAV5 vector, it has 10 years of exclusivity in marketing in Europe. And I should say in Europe, for adults in Europe means age 16. The USA FDA approved this therapy in June of 2023, again for adults with severe hemophilia without antibodies to AAV5. And in this case, the FDA required specifically an approved test for those antibodies. It's called a companion diagnostic. And so an official test was developed to measure AAV5. And I should point out in the USA, adults means 18 years old. So I think that's essentially what I want to present. But some final thoughts. It's clearly this is a substantial advance for hemophiliacs to be able to have one treatment and really not have to do weekly infusions. But the uptake in the market's actually been somewhat slow. And a big part of that is the marketing for this and a lot of other gene therapies is quite complex because it's a single treatment and you're trying to avoid what might be $500,000 a year over almost lifetime. So getting the marketing approved and reimbursement is not as straightforward as with normal drugs. A couple of other things have impacted this. That there are longer lasting proteins available now. So sometimes some of these proteins, they don't have to dose 2 or 3 times a week, but can be done once every 2 weeks. The safety of this is pretty acceptable, but some patients are still reasonably comfortable with their existing treatment. So I think it's taking a bit of time to get the marketing to going, but it will get there. And I think one concern for some people is that we still don't really know why the liver enzymes get elevated when they do. But nonetheless, and there, as I said, there are two products out there for hemophilia B from other companies, which are also beginning to start the market. And everybody faces about the same issues. There's clearly a lot of things for future study. One is, and this is, could we make more efficient vectors or better vectors, in which we could decrease the dose, avoid all liver enzymes. Some people still have questions about durability and how long does this really last after a single dose. Certainly the phase 1/2 subjects are out 5 years. In phase 3, it's clearly gone out more than 2 years. In fact, some of those patients are out 3 years. There's some variation in the level. It seems to go up high for a while and then decreases over time. The mechanism of that isn't really understood particularly well. There's a bunch of work being done on it. But nevertheless, the durability has been maintained. a second question is, could we dose those subjects who already have pre-existing immunity to the AAV capsid by using some sort of immunosuppression? And the same question would arise if you would need or want to redose patients who've been treated with the vector, because once they've been treated with that vector, they get substantial antibodies against AAV5. And a really important question is, this is treatment for adults, but the difficulty is that by the time hemophiliacs are adults, they've got a lot of the damage has occurred, and you would ideally like to treat very young patients, much younger patients, but their livers are still growing. And the issue there is that because the liver is growing and because an AAV vector sits in the cell, but it doesn't integrate into the genome, as the liver cells divide, you may lose the vector. So that's another set of issues which are being studied. But nonetheless, I think this is a really substantial advance. And I have to say, after 50 years of working on AAV, I mean, I personally think that it's nice to see that all of this technology and work has really gone to some clinical results. Finally, I want to show you one photo. This was taken in Oxford in 1987, and it shows the two guys who got me into AAV in the first place. You obviously will recognize Ken there in the middle. This is Jim Rose from NIH, also passed some time ago. And actually, it has in here too, the Florida people should recognize this is Nick Musichka, who also I passed away some time ago. So I look at this photo and it's, while these were people that had enormous impact for many years and we interacted with, it's a little bit nostalgic to just look at this and realize that all three have since passed. So I think that's all that I want to say. And I thank you for listening.
Questions? So, Barie, this was fascinating. How many patients have been treated, and the immunity that you mentioned, the antibodies to AAV, that's only seen in patients who've already been treated, so they can't be retreated. I mean, how
Yeah, do you mean how many patients for hemophilia or overall?
No, hemophilia.
Oh, I'm not actually sure what the number is. It's quite slow. It's a handful in Europe, and they're beginning to do patients in the US. One of the things that slowed it, the reason it's slow is in part too because this came on top of the COVID lockdown for number one, and secondly, having to then get these tests up and running all over. So it's still a quite small number. But certainly everybody in the clinical trials, everybody who's treated with the AAV5 vector will get a substantial antibody response against the AAV capsid, although in and of itself, that doesn't seem to mean much because the capsid, of course, goes away after the DNA is delivered to the cell. But it does mean, and this is true for any AAV vector, you can't just come around and give another dose if you didn't quite give enough. There are a bunch of studies going to ask, can you either inhibit that response initially by using some sort of immunosuppression, or can you block those antibodies after you've given the dose to see if you would want to give another dose. And that's sort of, there's an enormous number of people working on that, including I think people around at Gainesville. It hasn't got there yet.
Yeah. Okay. Thank you. Other questions? The questions? Okay. From the audience here and Zoom. Other questions? Madeline Mitchell. Which liver enzymes are elevated? Is that known?
Yeah, it's the ones that are measured ALT and I think some AST, so alanine aminotransferase and aspartate transferase. These are the standard liver enzymes, that you check for, in blood chemistry for normally looking for liver disease. But it's ALT and AST are the two that have primarily been measured and reported upon.
Okay. Now, when you treat with this gene therapy, is that factor then produced in the liver and not in the bone marrow? Where is it produced?
As far as we know, certainly for the AV5, virtually all of the vector goes to the liver and certainly is being produced in the liver. Certainly in the animal models, we can show that. I'm not sure how carefully the bone marrow has been looked at. The interesting phenomenon is that factor 9 for hemophilia B, we know that that's actually made in hepatocytes in the main liver cells and secreted. The issue with factor 8 is with this vector, it's also mostly being made in hepatocytes. But in fact, factor 8 is probably made in liver endothelial cells. So it's not being made in quite the cell it would normally be made in, although as far as we can tell, the protein that's produced looks exactly like the normal protein.
Yeah, that's fantastic. And then one other question is the cost. What is the cost to the patient or to the insurance company?
Well, that's the big debate because, as you know, there's all sorts of gene therapies out there at remarkable prices, and cell therapies as well. So I think, and I don't actually know what Barie has negotiated with all of these different insurance agencies. I think they suggested that in the US that the price might be 2.9 million. Their justification is that, well, if you the cost of treating hemophilia, either here or in Europe, is around about $500,000 a year, in some cases $1 million, because the protein is very expensive. And these patients are in and out of clinics almost weekly. Anyway, I think they said 2.9 million, but then what happens is, there's got to be all sorts of reimbursements and negotiations, and I don't think that that's the value that it's going to be. It's going to be substantially less. In Germany, I think it's something like 900,000 euros for the dose. But this is a sort of a debate. The issue is in Europe, there are single bursars, and so you have to negotiate with each individual company or each individual province in Germany. But it's a single bursars. The issue in the US, the sort of problem is that reimbursers here, you have your insurance for a year, but you might change next year, and so a lot of them are a bit wary at paying out all this amount of money for hemophilia this year when a person might switch next year. So these are just it's an ongoing issue for this and all other gene therapies as to what the prices really will be. And then there's been debates about, well, should you have to pay the whole amount, be reimbursed up front, or should you pay some amount per year based upon whether the vector is still expressing and the therapy is still working? You'd pay like 10% a year over 10 years or something. But I think pretty much all of it's being done as single payments at the moment. But I don't have, number one, I'm not even at Barie, and number two, pricing and selling of drugs is not anything I really know much about personally. So it would be great if it could be a lot cheaper. I mean, it's hard to believe that these therapies are that, but there are cell therapies, and there's a gene therapy for Zolgensma, which is, I think, being sold at $3.5 million a year. That's the one for spinal muscular atrophy.
Yes, last week we heard about that. Yeah.
Yeah, which I will tell you is absolutely dramatic, pretty much. But I think it's $3.5 million.
Wow.
Yeah.
And I have one question more. Where is hemophilia prevalent most, in Europe or here? Is it prevalent, found in all parts of the world?
Yeah, for hemophilia, I think so. I think it's pretty commonly prevalent pretty much everywhere. It's not like, thalassemia, which is another different blood disease. That, of course, is prevalent in Mediterranean countries in a few places, or sickle cell anemia, which, of course, gives resistance to malaria and is much more present in Black populations in Africa, etc.
Right.
I think hemophilia, it's pretty much commonly prevalent everywhere.
Yeah, because hemophilia in our mind is always the Tsar and the Russian.
That's right.
Guilty.
There's a photo of the Tsar's son, and he's it's often shown, he's standing in sort of a military uniform when he's about 14, but he's got one foot up on a stool. And the reason is that one of his knee joints was so mangled, he really barely could walk.
Oh, boy.
And it's been interesting. It showed on the slide. I'm not sure if you all picked it up. The Jewish Talmud actually describes an issue that says that if a woman has two sons and they both die after circumcision, from bleeding out, then if any subsequent sons do not need to be circumcised. So people knew about this long, long time ago.
Okay. Yeah, there's a question on, would you read the whole thing?
What gave the researchers the hope that with decades of work, they could overcome the challenging disease?
What
Yes,
kept us?
substantially overcome this challenging
Mhm.
disease. What gave you the hope to continue this work, in other words?
It's a good question. I think gene therapy started being talked about in the 1970s, early on. And particularly in the cases, very rare diseases. But I think two things, and particularly for the hemophilia, we knew that protein prophylaxis works. You can take purified factor VIII and you can treat hemophiliacs, and as long as you get enough of that in their blood, you can have a substantial impact on the disease. So the proof of principle for expressing that protein was there. What kept us going? Some of it was probably obstinacy. I think some of us who are not clinicians perhaps did not even appreciate early on just how difficult it is to develop clinically therapeutics, any sorts of therapeutics. Um, and I think otherwise it was partly it was an intellectual challenge too of solving the technology, but we just plugged away, a bunch of us. And I will say one thing, as you all know, Florida had the Gainesville group had enormous impact here because as a result of Ken's efforts, they developed so many people, hired so many people working on AAV that it was just, it just kept everybody going, and we would get around at meetings and sit around, we were talking. And I think I certainly will say for AV vectorology and I, with a particular parvovirus group, because we started off, when I started in around 1970, there were so few of us. There was Ken, there was Jim Rhodes, there was only about 3 or 4 labs. So we're a little group. We sort of had to talk to ourselves because nobody else cared about this virus that didn't cause a disease, didn't think it was relevant. I think that was infused through a large part of the field for a long time. And we have this thing called a parvovirus meeting, which is where that photo was taken, where everybody came aboard, all the graduate students, and people used to exchange data pretty freely. So I don't know. It's a hard question to answer, but maybe we didn't know any better.
I love this picture that you showed. I think Ken, the first thing he would have said was, "This is when he still had hair."
Yes. Well, exactly.
I'm sure
Yep.
he would say that, right, Laura?
Yeah, I know.
Okay, any other questions? Is there anything else on Zoom? Okay, thank you so much, Barie. We really appreciate it all the
Okay.
way from Seattle. Wonderful
Well,
talk. Great.
thank you very much.